Thank you so much for the invitation. It's been really lovely to talk to you all. I keep saying how well organised this all is. It's been really great to hear everybody's project today, and I look forward to more chats tomorrow as well. So I'm going to be talking about parallel evolution, and it's called Revealing the Silent Drivers of Extreme Parallel Evolution. So I am currently a PI at the University of Bath on a Royal Society George Hodgkin Research Fellowship. And just to give a bit of context, especially in this virtual world, as to where I am right now, I am at the University of Bath, which is just here in the southwest of the UK. It's kind of the gateway to the southwest. This area here is all very well known for its beautiful coastline and its countryside. It's a gorgeous city to walk around. It's a World Heritage City. The campus is slightly less gorgeous, the architecture, but it does a nice job, and it's a lovely place to be. And we're lucky to have, in 2018, a new building built for the evolution department of the Milner Centre for Evolution. So it's a good place to be, and when things open up, you should definitely come and visit. So very broadly, my research is involved in gene regulatory network evolution. So I'm going to start by giving a bit of an overview of that, and then talk about the model system that I use, which is experimental evolution with bacteria. And then I'm going to go into sort of a more recent project that's been going on in my lab, looking at the deterministic factors of repeatable evolution. And this is using the same gene regulatory network model system that I will have described previously, but I'll talk about how we've adapted it to look at this question of parallel evolution. And then I'll very quickly cover future challenges and directions before taking your questions. So yeah, very broadly, my lab focuses on gene regulatory network evolution. So just to begin with, I'll just talk about what gene regulatory networks are. So genes are often organised into groups that can be switched on or off together, and this is more efficient than controlling the expression of each gene individually. So the elements that regulate these groups of genes are known as the gene regulatory networks. So a more sort of formal definition might be that gene regulatory networks are a collection of molecular regulators that interact with each other and with substances in the cell to govern gene expression. And this is really important for determining function and therefore for determining cell fitness. So there are So in some ways, gene regulatory networks are very good at allowing an organism to change its phenotype based on environment. And a really nice example of that are two-component systems in bacteria. So these aren't present in eukaryotes, but they are a sense and response mechanism between the external cellular environment and the internal cellular environment. So what happens is that surface cell receptors might detect a change within the environment. This change is fed into a two-component system, which consists of a histidine kinase and a response regulator that will then initiate transcription of a certain set of genes that are important for fitness within this changed environment. So this will change the gene outputs, leading to a phenotypic change that can feed back and hopefully increase fitness in the environment. And so you can see that these gene regulatory networks are really important for fitness. So we know that they're important for fitness, but what do we know about how important they are for evolution in general? Well, we know that changes in gene regulatory networks are very important, and they're actually really common in evolution as well. We can tell this using bioinformatic approaches. So phenotypes can evolve very rapidly through changes in gene regulation, and that's because a single mutation within a regulatory region can have a very large effect. Whereas if you're hoping to get the same change by changing individual genes, you'd have to acquire multiple mutations at the same time. So we know that they happen, they lead to these large sort of phenotypic leaps, and they're common across all life, but being a human-centric species, we quite like thinking about our own evolutionary history. And so back in 1975, King and Wilson showed that there were actually these changes and evolution in gene regulatory regions were really important for our own evolutionary history. So if you look at the number of mutations within the genome between chimpanzees and humans, it was thought that there weren't enough differences to account for the phenotypic differences between these species. However, if you look at where these mutations are occurring and weight them based on the number of genes that these mutations are likely to have an effect on, then you can account for the number of differences between these species because a number of these mutations occur within regulatory regions. So we know they're important for fitness and we know that they're important for evolution. But what do we know about how these gene regulatory networks expand and adapt themselves? Well, this question is actually quite difficult, and this is because gene regulatory networks depend on a bit of a paradox. So their function depends on the specificity. So in order for these regulatory networks to be efficient, there needs to be a strong association between regulators and their target sites. And this is because if these groups of genes were going off at the wrong moment, they would have quite large fitness costs associated with it. So you want them to function efficiently. But in order to adapt, these networks rely on promiscuity. So in order for selection to be able to act on a new interaction within a network, it needs to be revealed to selection. And the way that they're revealed to selection is through promiscuous activity. So this is where a regulator might bind to a non-cognate target site. So you've kind of got these two conflicting mechanisms going on. So they need to be specific, but they also need to be promiscuous in order to allow them to adapt. So one way to imagine how this mechanism, a mechanism that might allow the expansion of gene regulatory networks whilst maintaining specificity, is through gene duplication and specialization. And so I've got a really simple example here. So if you're imagining that this is a progression over time, and in the ancestral state, you've got one transcription factor that is able to interact with both gene 1 and 2 to initiate transcription. However, over time, there's a random duplication event, and perhaps the selective environment is such that actually it's optimal to have gene 1 and gene 2 decoupled so that they're regulated separately. And so what you find here is actually divergence between these transcription factors, such that you get two independent transcription factors. It's transcription factor 1 that is able to initiate transcription of gene 1, and transcription factor 2 that's able to initiate transcription of gene 2. So these two are now regulated independently. But an artifact of this type of evolution is that you get built-in homology within these networks. So this is the ancestral state where you have the one transcription factor initiating transcription of both genes, and in the evolved state you've got two transcription factors. But because of this homology, the shared ancestral homology between these transcription factors, you still gain this weak binding affinity for their non-cognate target sites. And this can lead to opportunities for rewiring. And this crosstalk, this interaction with the non-cognate targets, has the potential to facilitate the evolution of novel regulatory function. And this is because it can reveal new interactions to selection, and this opens opportunities for rewiring. So it's really these opportunities for rewiring that I'm interested in exploring at the moment within my group. So in our lab, we study the evolution of gene regulatory networks through rewiring using experimental evolution with the bacterium Pseudomonas fluorescens. So Pseudomonas fluorescens is a very common lab bacterium. It's very well enumerated. It's fairly good at genetic modification. And it was isolated in 1989 from the leaf surface of a sugar beet plant grown in Oxford. So it's very well lab adapted as well. It's a gram-negative bacteria, and something that's very important for the work that we do is that it has these long flagella, these swimming tails coming out the back of the bacteria that help it swim along. This is what the wild-type bacteria looks like on a soft agar plate, and this is called swarming motility. And this motility is a product of both expression of the flagella and a biosurfactant gene called viscosin. And viscosin is like a slippery oily substance that the bacteria can excrete, and then it just allows the bacteria to slide over, reducing the friction and slide over a surface. So this is the wild type SBW25, and within this wild type you have FleQ, which is the master regulator of the Flagella network, and the Flagella network is built as a hierarchical network. So right at the top you've got FleQ. And FleQ initiates transcription of all the other Flagella and chemotaxis genes. So without FleQ, the cascade cannot begin, and therefore none of the Flagella genes get switched on. So FLQ sits right at the top. It initiates transcription of Flagella and chemotaxis genes, which leads to expression of a functional flagella and finally to a spreading motility phenotype. But in order to get this spreading motility phenotype, you also need to have expression of viscosin genes, either VisC-B or VisC, and this in concert leads to this particular pattern. So to start with, we engineered a non-motile or a FLQ deleted mutant. This is delta FLQ. So in this case, it's unable to express any of its flagella genes, which was confirmed via electron microscopy. But it is able to express its viscosin genes, and this is, the slippery biosurfactant. So there is a form of motility. It's called a spidery spreading phenotype, and this is a passive form of motility. So basically what they're doing is as they grow and divide, they're able to slide across the surface, but it's still a motility phenotype. So we wanted to remove all motility, so we also introduced a transposon into VisC-B, which knocked out viscosin production as well. And this is our non-motile mutant AR2, that if you put it in the middle of a soft agar plate, it's completely non-motile. And this can be confirmed via electron microscopy. However, what was actually unexpected, so we weren't planning for this, but this AR2 mutant was left on a plate over the weekend. And we noticed that what happened is it very rapidly evolved a slow spreading phenotype. So this is actively moving across the plate here. Under electron microscopy, we weren't able to see a flagella to begin with, although flagella expression has been confirmed with microarray analysis and video microscopy now. So it could be that either flagella expression within these slow spreaders is transient, so it's only switched on occasionally, or it's within a subpopulation. That's something we haven't actually confirmed yet. But anyway, so you have this slow spreading phenotype emerging very rapidly after about 48 hours, and then a faster spreading phenotype emerging after about 96 hours. And within this fast spreading phenotype, you can clearly see flagella expression on the electron microscope. So this was really exciting because, as I said previously, if you knock out FLQ completely, there is no way that it can initiate transcription of these Flagella and chemotaxis genes. So there must be something alternative taking on the role of FLQ, and at this point we were at a bit of a loss of what that could be. So the first step was to whole genome sequence the bacteria. And what we noticed is that there was mutations, 2 SNPs within 2 genes, ntrb and ntrc. ntrb and C are part of a 2-component system that's involved in nitrogen assimilation genes. So normally, ntrb and C, so within this non-motile mutant, what happens is ntrb is a histidine kinase that acts to phosphorylate ntrc. And ntrc in this phosphorylated form then goes on to initiate transcription of nitrogen assimilation genes. This first step mutation that we noticed was within the PAS domain of ntrb. And this led to the constitutive expression of ntrb such that it was always phosphorylating ntrc. This led to an increase of phosphorylated ntrc, a buildup of phosphorylated ntrc in the cell, and a massive increase of expression of nitrogen assimilation genes, so much so that it was actually toxic to the cell. And this is because it was basically bringing too much nitrogen into the cell. And what we also noticed was this weak interaction of phosphorylated ntrc with the Flagella and chemotaxis genes, such that it was able to initiate transcription of those genes. At this point, we didn't understand why, but the association was definitely there. The second step mutation was within ntrc, and it was within the helix-turn-helix domain. And so the effect of this second step mutation is to slightly compensate for this overexpression of nitrogen assimilation genes. So it reduced expression of nitrogen assimilation genes. So it reduced the toxic effect. And it also increased this link between ntrc and FLQ such that it was increasing Flagella expression. And so this was super interesting, but it left us with a really big question, which is, what has the nitrogen regulation system got to do with the Flagella pathway? So we confirmed sort of what was going on at the mechanistic level using microarray analysis. So this is looking across 2 different Go terms. So these are the genes associated with bacterial type flagellum. We also looked at genes associated with nitrogen compound transport. So the colour change matches expression levels. So warmer colours are higher expression, colder colours are low expression. And we're looking across isolates of SBW25, so this is the wild type. This is the delta flea Q strain. This is the non-motile mutant. And then you've got AR2S is the slow spreading mutant, and AR2F is the fast spreading mutant. And what you can see initially looking at the bacterial type flagellum genes sort of matches what we saw within our phenotypic work. But if you knock out flea Q, unexpectedly you get a massive reduction of bacterial type flagellum genes. This is consistent with an AR2, so introduction of the transposon into the viscosin gene doesn't have an effect on bacterial type flagellum gene expression. But this first step mutation within ntrb leads to an upregulation across all of the bacterial type flagellum genes, and the second step mutation in AR2F leads to further upregulation. And this is linked with changes in the nitrogen compound transport system. So within the wild type here, you can see just by knocking out flea Q, actually, we already see some slight reduced expression of some of the nitrogen compound transport genes, which suggests that there's already an interaction between these two networks existing. Nothing really changes within our AR2 strain. This is the one with the transposon and viscosin gene. However, this first step mutation within ntrb leads to a massive upregulation of many of the genes associated with the nitrogen compound transport. And the second step mutation in ntrc in the AR2F strains actually compensates. So some of these are actually reduced, some of these regions are reduced with lower expression. So this is compensating for the overexpression of the NTR system. And you can also see this phenotypically by growing them on different nitrogen sources. So this is looking at the doublings per hour. So this is like a growth rate of the non-motile strain, the slow spreader, and the fast spreader. So these bacteria were evolved on LB. So this is a complex media type. And you can see that through these mutations, they get better at growing on LB, which makes sense because this is the selective environment. However, if you look at them across different nitrogen environments. So this is a simple media where they only have access to glutamine as a nitrogen source, only have access to glutamate as a nitrogen source, or only have access to ammonium as a nitrogen source. You can see that typically they're quite capable of growing with ammonium as a nitrogen source. They're quite happy, and they're quite fit. However, with this mutation within ntrb that switches on the NTR system, they find that there's a massive cost associated with this, especially in the presence of ammonium, and this is due to the toxic effects associated. And this is slightly compensated for in this fast spreader, although it is still a costly mutation to have in these environments. So how is it that the NTR system is able to recover flea Q function? Well, this becomes slightly more obvious when you look at these proteins. So both of these proteins, this is flea Q, the one that was deleted, and this is NTRC, the protein that was co-opted to replace flea Q function. They're both sigma 54 dependent enhancement binding proteins, which means that they bind to the DNA upstream of the gene expression and interact with the sigma 54 factor to initiate transcription of genes. And in the absence of flea Q, NTRC is able to interact with flagellum assembly genes, and this is because they share a very high structural homology. So there's only about 30% sequence homology between these two proteins, but there's very, very high structural homology. And if you look at where the mutations are occurring within NTRC, they're all within this C-terminal DNA binding domain. So there's 3 domains within this protein. There's an N-terminal receiver domain, there's a sigma 54 interacting domain, and there's a C-terminal DNA binding domain. And it's this C-terminal domain that binds to the DNA and interacts with the DNA, the target sequence. So all of these mutations made NTRC more flea Q-like. So they increased the affinity of NTRC to binding sites for flea Q and decreased the affinity of NTRC for its typical cognate binding sites within the NTR pathway. So this now made sense, and it shows that the recovery of broken networks can occur through promiscuous activity of homologous proteins. And so this is the starting point for work that's currently ongoing in my lab. And the reason that this system works really well as a model system to explore the evolution of gene regulatory networks is that the phenotype that we're looking for is very easily observable. You're just looking for a motile zone on an agar plate. And we know that this motile zone can only be achieved by gene regulatory network rewiring, and that's because we've completely deleted flea Q, which is at the master regulator of the flagella network. It's also great because the evolutionary events are very rapid. They happen within a weekend, sort of within 48 to 96 hours, and they're very repeatable. So we know that whenever they happen, we are fairly confident as to which genes we need to look at to find the mutations conferring the phenotype. And we also have a very strong understanding of the genotype-phenotype relationship. Because we know that these mutations are likely to be occurring within the nitrogen pathway. We can ask questions like, what happens if you change the selective environment such that you make these mutations more costly? So sort of overarching aim of my research group is to understand the genetic and environmental drivers of gene regulatory network evolution. And I have 3 excellent PhD students that are taking this in slightly different directions. So at the top, we've got Louise Flanagan, who is looking at understanding the evolutionary and ecological consequences of gene regulatory network rewiring patterns. We also have Matthew, who's taking a more molecular and mechanistic approach. He's looking at defining the rules that determine gene regulatory network rewiring routes. And then James, who will be talking about his project in more detail. His was a sort of an initial side project that's taken on a life of its own that's really, really interesting. And he's been looking at the deterministic factors of parallel evolution. So this paper is currently in review and it's available on bioRxiv, and it's called A Mutational Hotspot That Determines Highly Repeatable Evolution Can Be Built and Broken by Silent Genetic Changes. Oh, wrong way. So I know a lot of you are already researching parallel evolution, so you don't really need me to tell you about this, but parallel evolution is very common in experimental evolution, and that's because you're working under very constrained evolutionary conditions. So you're basically, especially with bacteria, usually starting with clonal populations, and you are exposing them to constrained selective pressures, meaning that you expect a certain level of plurality within your independent lines. And parallel evolution just means the same evolutionary outcome across independent lines. However, the likelihood of parallel evolution really depends on the biological level that you're interested in. So parallel evolution can occur at the phenotype level. That's where everything at the end point of your experiment looks the same. Parallel evolution can occur at the gene network level, so that's where the mutations that confer this particular phenotype are occurring all within the same gene network, so for example the nitrogen regulation pathway. They can be parallel at the locus level, so you can find the same gene repeatedly mutating to confer this phenotype. And rarely we find that evolution can be parallel to the nucleotide levels. That's where you find the same mutation conferring this phenotype every single time. And this is actually really rare, and that's because typically there are multiple mutations that can confer a phenotype. And so probability tells you that if you run this experiment again, you would expect to see a certain frequency of all the possible mutations arising unless there's some bias occurring. So because we were interested in using this model system in our lab, we ended up repeating the process again and again, whereby we were starting off with these non-motile or low motile strains, allowing them to evolve motility and then genotyping, genosequencing the bacteria at the end. So what we started doing was growing these bacteria across different media sources. So this is LB, this is a complex media. M9 is a simple media where ammonium is the only nitrogen source available. And then we have M9 supplemented with glutamate or glutamine. The reason we started looking at delta flea Q as well is because although it has this spidery spreading phenotype on LB, within the absence of these complex nutrients, it's unable to access its viscosin expression, and therefore it ends up becoming a non-motile phenotype. And so we wanted to see whether there was any interaction with this transposon within this background. And all of these are evolved on a soft agar environment. So that's about 0.25% agar, which is quite liquidy, but it just means that it allows this flagella and viscosin system to work together. So what did we see when we did this a lot of times? So this is out of 116 independent replicates. And the y-axis here, you can see the proportion of mutant lines. So this is within each of these backgrounds, the proportion of different mutations conferring the motile phenotype. This is looking across different media types. So you've got LB, which is the complex media, M9, which is the minimal media, where ammonium is the only nitrogen source, and then you've got M9 supplemented with glutamine or glutamate. And this is looking at the strain delta Fli Q and AR2. And then the different colours confer the different mutations that were present within each of these motile mutants that we sequenced. And overwhelmingly, hopefully what you can see is that one colour dominates. And this is the mutation within an ntrb gene, A289C. And this is the mutation that we also picked up in the earlier work that I was discussing as part of my postdoc. So what we were expecting to see actually is that the media type might be having an effect on the different mutations that we see, because as I've shown previously, we know that this mutation here has a cost associated with it, especially when ammonium is the only nitrogen source present. But actually you can see that ntrb A289C is incredibly persistent across all these different media types and across strains. So this really grabbed James's attention, and he was really interested in delving deeper and finding what could be the drivers for this extreme parallel evolution. So he came up with three hypotheses. So firstly, it could be that there are very strong constraints, so that there are no other mutational routes available, that this ntrb A289C mutation is the only mutation that confer this phenotype. The second hypothesis is that natural selection is working very efficiently. So perhaps the most frequently occurring mutation is the fittest, and it's the best one available. And the third hypothesis is that there's some bias going on. So it's just that the common mutation that we're seeing is just likely to emerge first, or more likely to emerge than any other. And that's due to some underlying bias. So the first hypothesis, that there are no other alternative routes available, can be rejected straight away, because we can see there are other mutational routes available, although they're just occurring very rarely. And as I pointed out before, you can also see that they are independent of media type. So it's not that certain media types are selecting for different mutants. It seems to be that for whatever reason, this ntrb A289C gene is overwhelmingly dominating the population in all of the evolved strains that we sampled. So the second hypothesis is that the common mutation is the fittest mutation, and that seems sensible. So on the y-axis here, you're looking at the relative migration area over 48 hours. So this is just measuring the area of spread. This is looking across the different media types, M9, M9 plus glutamate, M9 plus glutamine, and LB. This is looking at the different mutation types. So for this analysis, we grouped together ntrb delta 406, 417 and ntrb delta 410, 421, because the transcribed protein of these is exactly the same. It basically deletes the bases that equate to the same amino acids. So we group those together. So we're looking across 4 different mutants, and it's important to remember this is relative to the A289C mutant. So the A289C is the baseline at 0, and then anything below this means it's moving slower than the common A289C mutant, and anything above it means that it's moving faster. So immediately what we can see is this GLUN-K, which is a different gene, but still within the nitrogen regulation pathway. It's actually the repressor of ntrb. So the mutation within at site 84 in GLUN-K is significantly less fit than A289C. So we can actually say, fine, we wouldn't expect to see this very frequently within our evolved populations. However, the other mutations are more difficult to explain, because statistically there is actually no difference between these alternative mutations and A289C. We can say, however, that because these deletions involved more bases, that we would expect them by probability to occur at a lower rate. And this is because if you're just changing one point mutation versus lots of point mutations, you would just expect this to happen less frequently. But what it does cast a big question mark over is this mutation here. So this is mutation A689C, also within the ntrb gene. So it's an A to C transversion. It's within the ntrb gene, and it's just a few bases downstream. So given that we can't statistically differentiate fitness of this mutant from ntrb A289C, we would expect to see this at a higher frequency within our evolved populations, but we don't. So the third hypothesis that we wanted to test was whether or not the common mutation might be the first to emerge due to some underlying bias. So to test this, we looked at, so the y-axis here is looking at the establishment on the frontier. So this basically means which bacterial strain would reach the leading edge first when they're in competition. So it's like what we call a race assay. So we co-inoculate them into the center of the plate at the same time, we let them race, and then we see which mutant is present at the leading edge. So this is in 3 out of 4 replicates. But instead of always co-inoculating them at exactly the same time, what we did is we gave this A683, this less common mutant, a head start. So we either gave them a 3-hour head start, a 6-hour head start, 9 or 12. So this was just to test as sort of how far in advance do you have to gain this mutation to get a competitive advantage. So when they're co-inoculated at exactly the same time, in 3 out of 4 of the cases, you find that the A289C mutant dominates. So this is saying, okay, so we might expect, this has a small fitness advantage. So you would expect about a 75% maybe frequency within our evolved populations, but that's still a lot lower than what we're actually seeing. And then after 3 hours, you find that this relationship switches, so that in 3 out of 4 of the replicates, the A683C mutant reached the leading edge first. So this suggests that within a 3-hour window, that A683C is able to gain a competitive edge. And then after 6 hours, it consistently was always at the leading edge, this rarer mutant out of 4 of the replicates. So this suggests that actually there is a massive advantage from getting in there early. It's sort of the early bird catches the worm maxim. And this is saying that you gain a really strong competitive advantage of being the first mutant out the gate. And this also suggests that A289C is more likely to occur first. So there should be some underlying bias that means that this mutation, even though it's only a few bases upstream and it's the same A to C transversion, seems to be occurring first within these evolving populations. So what is it that's causing this bias? This is a complicated slide, so I'm not going to go through all these different mechanistic causes, but just to say that there are a number of mechanistic causes of local mutation bias. So what James did was he did his best to sort of filter out as many of these as possible. But really what we were looking at is something that could confer very localized mutation biases. So this is at a single nucleotide point. So this isn't within a range, this is at a single point. And so one particular mechanism jumped out to him that he wanted to investigate further, and this is number 7. So this is showing that alternative DNA secondary structure formations such as hairpins can impact local mutation frequency. So just to sort of help visualize this is sort of what happens. So within this DNA stretch here, there are 2 complementary inverted repeats here. And when the DNA comes apart for replication and moves into single-stranded strains, it gives the opportunity of these inverted sequences to come together and to form complementary strands. And so what happens is you get these hairpin loops. And what this can do is that as the DNA polymerase is traveling down the DNA and it hits these hairpins, it can basically trip and stumble over them. And this increases mutation rate, specifically at the base of these hairpin loops. So this is something that's been looked at a lot theoretically, but not too much empirically. And so there was an interesting tantalizing suggestion for a hairpin loop within around the region of site 289 within ntrb, but it wasn't enough to really be confident of anything. So we had to think, right, how could we go about testing this within our system? Well, luckily, we had another PhD student, Louise, working with a slightly different Pseudomonas fluorescens strain, which is PFO1. So SPW25 is the strain that I've been talking about so far that was isolated in Oxford in 1989. PFO1 is also Pseudomonas fluorescens, but it was isolated in Massachusetts in the US in 1991. And this strain, when you delete the FLEQ gene, master regulator Flagella network, you also get a non-motile strain. And when you leave it to evolve in a soft agar plate, rapidly you get a slow spreading mutant emerge. So we were at the time genotyping these mutants to see if there was any variation in what mutations that we saw. So first, what we had to think about was how similar is the ntrb gene in SBW25, the gene that we've been looking at, the strain we've been looking at previously, and PF01, this new strain that we're interested in. So we can say that ntrb genes are similar, but they're not identical. So this figure here shows the nucleotide sequence, and you can see that there's about 89% similarity between these sequences. And this shows the amino acid sequence, and there is approximately 96% sequence homology between these two strains. And highlighted in red here, so this is the threonine that is repeatedly mutated. It's mutated to a proline within the slow spreading mutants, and this is the adenine that's typically mutated to a cytosine, site 289. So if you look at the amino acid sequence, you can see that there's a very good match between the amino acid sequence immediately upstream and downstream of this threonine. So there doesn't seem to be a lot of variation at the amino acid level. However, if you look at the nucleotide level, whenever you see a gap here, that means that there's a mismatch between these two strains, and you can see that there are some differences. So these are synonymous mutations, so mutations that are confer a difference at the nucleotide level, but they are not translated into any differences at the amino acid level. So this gave us a nice mechanism to explore whether or not these differences might confer a difference in mutation variation within our evolved populations. So this is what you see within PF02X, so this is the non-motile PF01 strain. Again, looking at the proportion of mutant lines across, this is two different media types, LB, the complex media, and M9, the simple media with ammonium as the only nitrogen source. And every colour here correlates to a different mutation type. So there's two things to note here. So firstly, there's a huge amount of variation in the different mutations that we're seeing, but importantly, they're all within the same network, so they're all within the NTR system. So they're using the same mechanism to confer this slow spreading phenotype. So they're still, all of these mutations lead to hyperphosphorylation of NTRC, that then means, and then the second step mutations are still within the NTRC gene. But you can see that there isn't this bias towards A289C that we're seeing within AR2 strains. And importantly, we've never seen yet this NTRB A289C mutation occur de novo within PF02X. However, we have engineered this mutation within a PF02X background, and it is viable, but we've never seen it evolve de novo. So this was nice because it gave us a system where we could empirically test the impact of these few synonymous mutational changes on the predictability of evolution at that site. So what James did is he mutated 6 bases upstream and 3 bases upstream and 3 bases downstream. So within a 15 base pair flanking region of site 289 to try and disrupt this hairpin structure and relax the structure into so that it didn't form these hairpins. And interestingly, so these are the sites that he mutated, just say 3 upstream and 3 downstream. So this is what happens in AR2 within LB and within M9. I've shown you previously. Overwhelmingly, you find that the mutation that dominates is A289C. However, within the AR2 synonymous mutant, you see a much wider range of mutants accessed within our evolved populations. And really interestingly, we have never seen a NTRB A289C mutation occurring de novo, although again, we've engineered it into this strain, into this background, and it is viable. So what we've shown here is that parallel evolution at site 289 in M9 fell from more than 95% to 0%. We've never seen the same mutation popping up twice within this background at the same site. So we've successfully broken a mutational hotspot. But if we can break a mutational hotspot, does that mean we can build a mutational hotspot by doing the converse experiment and mutating the same sites within PFO2X, but to be more AR2-like, so to change them to the equivalent AR2 sequence. And so that's exactly what James did. And indeed, it does work that way around. So this is our PFO2X synonymous mutant with the 6 synonymous sites mutated. And you find that parallel evolution within M9 rose from 0% to 80%. And really importantly, all of the mutations, the overwhelmingly most common mutation that we saw was this ntrb A289C mutation. And this is something that we'd never seen evolve de novo in the PFO2X strain. So this suggests that yes, you can successfully break a mutational hotspot and you can successfully build a mutational hotspot through very few mutational changes. And importantly, they're synonymous. So these effects are usually disregarded because they're considered not to be important to adaptive evolution because they don't show up in the protein sequence. However, here we've shown that actually they are determining the parallel trajectory of evolution within this strain. And this can also be backed up, this effect can be backed up looking at the phenotype time to emergence data. So on the y-axis here, you're looking at the proportion, this is just histograms, proportion of mutant lines in which a motility phenotype emerged across a certain day, up to 2 weeks is what we allowed. This is looking in LB and in M9. The dark grey strains are AR2 and the white bars, sorry, are AR2 synonymous mutant. So as I said previously, when we were looking at the sort of co-inoculation data, it suggested that you needed this A289C mutation to be occurring more quickly in order for it to be competitive. And so if you look at when phenotype or these motile mutants emerge within AR2, it tends to be around 2 or 3 days. And that's in comparison to the AR2SM mutant, where suddenly everything gets shifted to the right. And we're actually seeing that these motile mutants emerge sort of 7 to 8 or even 10 to 11 days. So there's a real shift to the right here, so that the phenotype emergence is happening much later, and that suggests that this is because the mutational hotspot has been broken, and therefore these mutations aren't as accessible as early. And this showed the same in PFO2X. So the dark grey bars are the PFO2X, and the white bars are the synonymous mutant that's more AR2-like. And you get the converse relationship here. So in the synonymous mutant, where you've built this mutational hotspot, you get a shift to the left where motile mutants are occurring more quickly and earlier than in the PFO2X. And that's across both environments. Okay, so why is this important and why should we care? Well, really it challenges our understanding of the role that silent mutations play in adaptation. So typically you often hear that we disregarded all the non-synonymous mutations because they're not expected to be contributing to the adaptive process. And here we show that actually they can be contributing and it's important to at least acknowledge them. It really raises the question about how often mutation biases might be biasing adaptive outcomes. Again, within experimental evolution studies, when we see these strong signatures of parallel evolution, it's assumed to be due to strong selection and fitness effect. And while this may be true, we really haven't done much exploring about whether or not there are lots of biases or whether biases are occurring that might actually be the underlying driver of these parallel events. And it also highlights the role of molecular factors in driving predictable evolution events. So forecasting evolution and predictable evolution is becoming quite trendy within evolutionary biology as a research area. But then we have a lot to understand at the molecular level as to really what are the players and what are the drivers that might be facilitating parallel evolution. And so it really begs the question that we need to go in and see if we can find evidence for potentially secondary structures contributing to parallel evolution more widely. So within my lab, our current immediate focuses are trying to explore a little bit more about what role genome position is playing in the deterministic activity of a mutational hotspot. So I showed you quite a complex figure about all the different factors that can contribute to mutation heterogeneity across the genome. And so we're playing about with moving this mutational hotspot to different regions closer to the origin, or on the lagging versus the leading strand, and trying to understand what that means in terms of its activity as a driver of parallel evolution. We're also looking at the evolutionary consequences of mutational hotspots in evolving populations. So we have now one strain that evolves very predictably and also very quickly, and one strain that evolves less predictably and slower. So it's a bit like a tortoise and a hare model. So really what we're asking is it better to be the tortoise or better to be the hare? Because the tortoise is able to explore more of its mutational space, potentially accessing a higher fitness peak, but the mutations occur slower. So you've got the hare that will always access the same mutation that's pretty good, it's fairly fit. And so within a competitive environment, which one is likely to win out? And then finally, we're looking more at the sort of mechanism and the molecular level of understanding the rule book for rewiring gene regulatory networks. So can you predict which regulators are likely to be co-opted to recover function? Is it as simple as the most homologous regulator will always be recruited? What about patterns of expression and underlying gene regulatory network architecture? So we're trying to sort of build all these factors in to understand what might be determining rewiring patterns and whether or not they can become predictable. So it's really, really important that I acknowledge the people that did the work and collected the data. So that's James Horton and Louise Flanagan collected all the data that I talked about in this talk. Matthew Shepherd is looking at the determining the rules for gene regulatory network evolution. Josie is looking at the evolution of CRISPR-Cas systems, and she's just joined my lab doing some really exciting work. And Aidan is our brilliant research assistant, and Nick, Rob and Louise have all been really influential at different points of this project, and it's been funded by the BBSRC and the Royal Society. And if anybody's interested in sort of following the development of this story, you can follow me on Twitter. And I think that's my time, so I'll happily take any questions.